Okay, so let me talk to you about what you need to understand about glycolysis. I think you'll be happy because it's a lot less than you might need to. For example, I don't need you to know any of the specific reactions. I'm basically going big picture here, in and out, why, that kind of thing. So why? So this is the point of glycolysis. So one part is you start extracting electrons from your carbon compound. If you're going to go through glycolysis entirely, it has to be starting with glucose. So start extracting those electrons, which is like overall the point of cellular respiration. And then the other point of cellular respiration, make ATP. Now you only make a little ATP here. Make a little ATP. That's the point of glycolysis. We're going to notice to extract those electrons, we have this really important concept here, is that if you, to extract electrons from a molecule, you basically break a bond. So you break a bond, and that allows you to extract electrons. And by you, of course, I mean the enzymes. So break a bond, extract electrons. And you'll see this over and over again. Break a bond, extract electrons. Break a bond, extract electrons. This process, unlike the other parts of cellular respiration, happens in the cytosol. So it's not mitochondria dependent. It's a very ancient process. And it's also not oxygen dependent. It's used in both cellular respiration, that's aerobic, needs oxygen, and glycolysis is also a process used in fermentation. And it can be used by organisms with mitochondria and without mitochondria. So like prokaryotes and eukaryotes. But only some do it. Okay. Let's see what you need to know. So I'm just drawing this very schematically. Glucose would actually be in a ring structure. You don't need to know that. You just have six carbons, right, in your glucose. C6H12O6. So you've got glucose, and the glycolysis process is going to break that molecule. So glycolysis, glyco is like sugar, lysis is breaking, just like hydrolysis, breaking reactions, and lysosome, the organelle where breaking happens, like as in breakdown, breaking. So sugar breaking. So let's break a bond. And then what do we say we can do? Break a bond, extract electrons. So you take some electrons out. How does that happen? You have to have an electron shuttle or electron carrier. So this NAD plus, the plus here indicates it's without the electrons. So it's empty. Like if you've seen a taxi cab, if it has a light on, it means that it doesn't have passengers, so you can hail it. And if it has light off, it means it already has passengers. So you can think of electrons as passengers. Okay, so we have our NAD plus come in and then pick up electrons, picks up 2 at a time, and then that picks up hydrogen. But I'm worried about the electrons. But then it becomes NADH with the electrons in it. And that's going to go to the electron transport chain if you're doing cellular respiration with glycolysis. Let's say it like this, or the NADPH or the NADH can go to fermentation, as in undergo fermentation process. Okay, so extracted electrons, broke a bond. Now the thing is here is that the bond is broken, but there's not anything released. No, there's electrons released, but no atoms are released. So it ends up just, instead of 1 6-carbon, it makes 2 3-carbon compounds. Let's fix that. They're called pyruvate or pyruvate, slightly different emphasis. two of them. So you still have your 6 carbons. So there's no CO2 being made here. Not by glycolysis. Let's make a little ATP. So ADP plus, and I'll show you what this means in a second. And we just represent it with a triangle and a phosphate group attached to that. And that comes out as ATP a little bit. I don't need you to know numbers, just know it's a little for glycolysis. So the triangle is just representing a substrate that phosphate is attached to. So at some point, a phosphate came in, was attached to the substrate, which is just the molecule. Substrate meaning, molecule that's going to bind the enzyme. And then in this step here, with ADP, basically you end up transferring that phosphate onto the diphosphate, so you end up with that substrate that's dephosphorylated, meaning the phosphate's gone, and then the ATP. Do cellular work, and then this will be recycled back to get more phosphates on it. This kind of ATP production is called substrate level phosphorylation. Remember, phosphorylation just means adding phosphate. And the thing to realize about substrate level phosphorylation, which also occurs in the citric acid cycle, as far as ATP making, it just doesn't make a lot in the process. So you only make a little ATP. For some cells, that's enough. Yeast can just make ATP from glycolysis and be okay. But like for our cells, it's not enough. You can't just do glycolysis. You have to have the cellular respiration part, the rest of it with the mitochondria. So in the end, we have NADH carrying electrons away, we have 2 pyruvates, and we have a little bit of ATP for the cell to use. And that is what you need to know about glycolysis.